everyone. This is Clement from the University of Texas at Tyler. I'm very glad to have this opportunity to share my work with you. And also, I would like to sincerely thank ASBMB for organizing these webinars so that at this time we can still have a platform to share our work and communicate with other scientists. So here, let me switch to my PowerPoint presentation. Let's see. All right. Okay, so today I'm going to talk about how we can use a module swapping strategy to engineer some proteins. They are called allosterically regulated transcriptional regulators, and they can use for actually controlling gene expression. So in this talk, there are two parts. The first part, I'm going to show you how we can robustly engineer these regulators. And in the second part of this talk, I'm going to show you how we can use these engineered regulators to establish some new cellular behaviors that cannot be accessible previously. Okay, so the first part of this talk is to develop some hybrid transcriptional regulators with swappable functions in small molecule sensing and promoter recognitions. So there are lots of terms here, but don't worry, in the next 3 minutes, I'm going to show you what exactly that means. So for some transcriptional regulators, they have highly conserved structures and functions. So these regulators, for example, here as shown on this slide, this is from a family called LAC-I. So all these proteins has N-terminal DNA binding domain that bind to specific sequence of DNA. We call them operator. And also they have C-terminal regulatory domains that bind to specific small molecules. We call these small molecules effectors because the presence of these small molecules can affect the binding affinity of the protein to the DNA. So these proteins actually control gene expression, and their functions and mechanisms is also quite simple. So these regulators, they physically bind to the DNA, which then blocks the RNA polymerase to approach the DNA. But then with the presence of a small molecules, such as an inducer, then these proteins detach from the DNA, which then allows the RNA polymerase to bind to the DNA to initiate transcription, which then lead to gene expression. So you can see that the system is very simple. Basically, there's only one protein, and with one protein, the cells can detect a specific small molecules, and based on that to control the expressions of genes. So scientists have discovered a wide range of transcriptional regulators, and they can sense many different small molecules, from something as simple as a single metal ions like sodium ions and zinc ions to some more complex molecules such as fatty acids. So one problem of using these regulators to control gene expression is that they are not standardized and also they are not modular. So here, let me try to use this slide to explain to you what I mean not modular. So here, for example, we have two systems. So regulator A control gene expression driven by promoter A is response to the small molecule A as an inducer. And this regulator B here control gene expression driven by promoter B and is response to the small molecule B as an inducer. So by using these two regulators, we can then control gene expression driven by promoter A by using small molecule A. And then we can also connect small molecule B or effector B to promoter B. But then these connections are rigid and we cannot change them. For example, we cannot use small molecule A to control gene expression driven by promoter B. So this is what I call being not modular. And this actually provides some boundaries or like some barriers for us to develop new like genetic pathways for cellular response. Okay, so to deal with this problem, we propose to use a module swapping strategy to build some hybrid transcriptional regulators which can be used as connectors between different signals and different promoters. So here, for example, if we can fuse a ligand binding module of protein A or regulator A to the DNA binding modules of protein B, then we can generate a hybrid regulators that can control gene expression driven by promoter B, and then it can respond to small molecule A as an inducer. So basically, this is a new connection. And by doing that, we can then connect the signal A to promoter B. All right, so this is our goal. And to reach this goal, we first need to understand the structures of these regulators. And we first study those proteins in the LAC-I family. So all these regulators in the LAC-I families are highly conserved. So they have a regulatory domain, this green part here, it has a hinge helix, this orange part here, and also has a helix, turn helix motif that binds to the DNA. So from this figure here, you can see that the helix, turn helix binds to the major groove of the DNA, and it directly interacts with the nuclear bases of the DNA. And for the hinge helix, it also binds to the DNA, but it only binds to the backbone of the DNA. And so these interactions is not as specific as the interactions between the helix, turn helix and the DNA. And you can see that this structure is highly conserved. For example, I'm also showing a structure from another protein called purine. If you look at these two structures, they are highly similar. And also some previous NMR studies shown that when the proteins binds to the DNA or dissociate from the DNA, only the structure of the hinge helix is changing. So it shows that basically the hinge helix is involved in controlling the protein structures, which then mediates the binding and dissociation of the protein from the DNA. So based on these previous studies and also some other bioinformatics analysis that we have performed, we come up with the model on the right-hand side of this slide. So we believe that the helix, turn helix motif is for recognizing the DNA sequence, and the hinge helix is to control the protein conformation. So when there's a small molecule effector binds to the regulatory domains, it triggers a change in the conformation of the hinge helix. Now with this new conformation of the hinge helix, the helix, turn helix motif moves to orientation that does not favor its binding to the DNA, and so the protein dissociates from the DNA. So if this hypothesis is correct, then potentially we can change the DNA binding specificity of the protein by just swapping the helix, turn helix motif. And so we try to test this hypothesis. The first thing we have done is to build a LAC-I Gal-Al hybrid regulators. So here we fuse the LAC-I DNA binding domain to the Gal-Al small molecule binding domain to generate this hybrid protein. And we then use these hybrid regulators to control the expressions of a GFP gene in some E. coli cells that is driven by the LAC promoter. And you can see that here, when on the right-hand side of this slide, you can see that when cells containing this system is exposed to galactose, there's a dose-dependent increase in expressions of the GFP. So this result shows that the LAC-I Gal hybrid regulators can control gene expression driven by the LAC promoter, which is the properties from LAC-I, and then it can respond to galactose as an inducer, which is the properties from Gal-Al. So it shows that our hypothesis is correct and our strategy is working. And by building these regulators, we can then now build a new connections between galactose and the LAC promoter. So we have extended strategies to a number of other proteins. So on the right-hand side here, it shows 40 proteins that we have built by using 5 small molecule binding modules and 8 DNA binding modules. And of course, in these 40 regulators, actually 5 of them are native proteins because like both the ligand binding modules and the DNA binding modules are from the same original protein. And here you can see that for these 40 regulators, many of them are working very well. Specifically, 22 of them can generate more than 10-fold of increase in the expressions of GFP when they are induced by the desirable inducer. So we can potentially use them to build like some genetic networks that can lead to new cellular functions. All right, so now we have a set of hybrid regulators that are functional. So the most frequent question we get is actually why we want to build these regulators. What can they be used? So to demonstrate that, in the next part of this talk, I'm going to show you how we can use these regulators to develop a system. We call it the multiple toggle switches with a master of signal. So first, I would like to introduce what is a toggle switch. Many of you may not heard of that or may not be very familiar with a toggle switch, but probably you have used one in your daily life. So a light switch is a toggle switch. So for a toggle switch, it can detect basically 2 different signals, and each of the signals can then move the system to a stable state. So in the case of a light switch, the 2 stable states would be lights on and lights off. And because of these properties, a toggle switch can also be used as a memory device to remember the last signal that it has received. So a light switch is a toggle switch, but in biological systems, we can also build some genetic toggle switches. So here on this slide, it shows a toggle switch that we have designed and built. So in this toggle switch, it has two regulators, which are TET-out and RBS-out. So these two regulators, they repress the expressions of each other. And also TET-out control the expressions of a GFP gene, which is a reporter gene. And RBS-out control the expressions of mTOR genes, which is another reporter genes, which would make the cells become red. So with this system, we can then develop the toggle switch behavior that I just mentioned. So for example, here you can see that when the cells are exposed to one of the signal, which is ATC on the left-hand side here, you can see that exposing cells to ATC will lead it to move to the green state. And even after the cells disconnected from the signal, it will continue to be in the green state. And when the cells are exposed to ribose, which is the other input, then the cells will switch from the green state to the red state. So basically, this is the toggle switch behavior. So a toggle switch can potentially be very useful. So some scientists have proposed to actually build multiple toggle switch in some engineered bacteria, and then use this engineered bacteria to detect multiple physiological conditions inside a host. So for example, if the cells are being put into a mouse, then it can then test actually if the mouse has like an increase in like certain metabolites, or if there's the presence of different pathogens. But then one problem for using these multiple toggle switches is that based on the current design, each of the toggle switch, if we want to reset it to the off state, then it needs to be exposed to a different biological signal. So by using so many biological signals to reset all the toggle switch can be problematic. It can lead to some allergic effects, or it can be very inefficient for actually reset all the toggle switches. So to deal with this problem, here we propose to build multiple toggle switches with a master off signal, meaning that only one off signal is needed to reset all the toggle switches. So as I described earlier, for those hybrid regulators that we have developed, they can actually control gene expression driven by different promoters, but then they can respond to the same small molecule as an inducer. So here, for example, we have two regulators shown on this slide. Both of them can respond to ribose as an inducer, but each of them responds control gene expression driven by a different promoter. So by using these two regulators, we then build two different toggle switches. So for the toggle switch on the top, when it is exposed to IPTG, then it would turn on the expressions of an mTOR gene, which is the reporter. And when for the toggle switch at the bottom, when this is exposed to ATC, then this would turn on the expressions of GFP. And then when these two toggle switches, when they are exposed to ribose, then both of them will switch back to the off state. So with this system, we can then achieve this multiple toggle switch behavior, but then with one master off signal. And this is what we have built. So we built these two toggle switches in E. coli cells. And here you can see that when the E. coli cells are exposed to IPTG, it's become red. When these cells are exposed to ATC, it's become green. And when the cells are exposed to both IPTG and ATC, then it's become both red and green. But under all these conditions, when the cells are exposed to ribose, which is the off signal, then they all being reset and go back to the off state. So it shows that our idea is working. And by using these hybrid regulators, we can then achieve this new function and new behavior in a cellular system. So again, like by using this system, we can then more easily to build like a biomonitoring device that can monitor multiple physiological conditions inside a host. And by using only one inducer to reset this, all these toggle switches, then it is less likely to develop any adverse or allergic effects in the host. And also it would be more efficient for us to reset all these toggle switches, because now we need to only worry about whether this one signal can achieve these engineered cells in a complex host system. Okay, so here this is spotlight sessions. So I have like limited amount of time to talk about my work here. So if you would like to know more about this work, you can reference to these two papers that we published last year. And at the very end, I would like to thank all the people who have contributed to this research. So here are some of the members of my current lab. And also, I would like to thank my collaborators at UT Tylers, including Professor Joe Clayfie and also Professor Dustin Patterson. And also, I would like to thank my collaborators at UT Dallas, including Professor Farouk Mokos and also his former grad students, Shan Li. In terms of financial support, here I would like to thank all those agencies who have supported us. So the project is currently supported by National Science Foundation through the MCB program. And also we receive funding from UT Tyler as well as the University of Texas System. At the very end, I would like to thank for your attention. And this is all I'd like to share, and I'm happy to take questions.
Thank you, Clement. That was really interesting work. You've got a number of questions that I would that have been posted that I will read to you. The first one actually is how do you define the difference between a biologically active regulator versus an efficient regulator?
Yeah, so like it is kind of arbitrary and it is totally empirical. So from our experience, if a regulator can generate more than 10-fold increase in the gene expression, so this is based on the dynamic range in response to the specific inducer, we think it is actually efficient. And this is actually supported by the toggle switches that I've built. So by using these regulators, we can actually build a new behavior. I think building the new behavior is the most stringent test to show that these regulators are efficient.
And can the toggle switch be designed to be concentration dependent? What are your thoughts on that?
Yeah, I think this is a very good question. I guess if we can engineer the sensitivity of those sensors, potentially we can actually change the sensitivity of the toggle switch. So obviously right now what we do is we just use the concentration that can be detected by the regulators. But as a next phase of this research, we try to use computational methods, try to redesign the binding sites for these effectors. So potentially we can actually modulate the sensitivity of these regulators so that it can actually change the dynamic range of sensing as well.
And then another question is, would your method work for circularly permuted proteins as well, or is it just specific to the module switching you discussed?
So, you know, like right now, we have tested like several different protein families. So here in this talk, I focus on LAC-I, but we have also tested some other regulators families. So it seems that like some other regulators families also works. And actually last year is a good year. Like I almost want to call it the year of like module swapping of regulators, because besides my lab, like at least 2 other labs has published papers by using this kind of module swapping in other protein families. And seems like this module swapping approach is very robust, and it actually can be extended to many different protein families.
Okay, well, I think we'll stop the questions at that point and move on, although there are other questions, Clement, that I'd encourage you to answer directly in the Q&A. Thank you, everyone, for your questions, and thank you for the very nice talk.
Thank you, Blake. Yeah, so I will definitely go in and answer other questions. So feel free to keep sending questions if you have any for my talk. Okay, so now let me introduce the next speaker. So the next speaker is Nipuna Weerasinghe. So he's from University of Arizona, and his talk is modulation of GPCR function by membrane lipids and water. So here, let me hand the time to Nipuna. So Nipuna, please go ahead.
Thanks, Clement. Good morning, everyone. So I would, first of all, I would like to thank ASBMB to provide me this opportunity to present my research work. And I would like to start my talk. So today I'm going to briefly discuss the research work we conduct at Brown Lab to determine the effect of soft matter, which is primarily membrane lipids and water on the activation and modulation of GPCR function. So here is my content. So first of all, I will briefly discuss why we want to study GPCR. Then I will explain the experiment we did, the results we obtained, and the conclusion we made on the role of cellular water on the rhodopsin activation process. Then finally, I will present a new signal transduction model we developed to explain our experimental results. So GPCR constitute the largest superfamily of cell membrane proteins in our genome. And they are primarily responsible for transduction of extracellular signal into the cells and initiation of the downstream signaling cascade by activating G proteins. So currently, about 30% of drugs available in the market exert their activity through GPCR, and these drugs account for around $180 billion of sales globally. However, all these drugs only target around 50 GPCR out of more than 800 GPCR in our genome. And therefore, there is a huge potential to target more new GPCR and also improve the efficacy of already available drug through understanding the structure-function relationship of GPCR. So in our group, we use rhodopsin as a model system to study how cellular soft matter affect the GPCR activation process. So rhodopsin is responsible for demyelinized fission in vertebrates, and they are located in the outer membrane segments of rod cell in our retinas. During the activation process, the chromophore retinal in 11 cis form is photoisomerized into all transform, and this triggers a cascade of conformational change and establish a dynamic equilibrium between metarhodopsin 1 and metarhodopsin 2 conformational state. These metarhodopsin 1 actually have no ability to activate G protein. However, metarhodopsin 2 is the active form that can activate the G protein transducing. So according to the results available in literature, especially based on the structural method like crystallography, during the activation process, the protein dehydrate. As you can see, so the first picture is dark state rhodopsin, which has more cellular water. So these blue spheres indicate cellular water. And the second picture is representing the meta 2 rhodopsin, which have a less number of water molecules in the structure. But you also have to remember that X-ray crystallography technique obtain these structures under extremely dehydrating conditions. So they may be actually more like a fossil rather than actual protein structure in the physiologically relevant state. So, but according to molecular dynamics simulations, during the activation process of rhodopsin, there is an influx of around 60 molecules of this directly contradict the X-ray crystallographic data we have just seen. So this is another like a few snapshot from that molecular dynamic simulation. The first top left picture represent the dark state rhodopsin. Bottom left represent the meta 1 state, and the bottom right represent the meta 2 state. So as you can see from these pictures, meta 2 is highly hydrated than the meta 1 state. So this is contradicting the X-ray crystallographic data. So to address these inconsistencies, we carry out set of experiment by using rhodopsin in their native disc membrane and under different osmotic pressure conditions. Our assumption is that when we have a different osmotic pressure, it will favor the more dehydrated conformation. And according to what we already talked, so M1 is more dehydrated state, so higher osmotic stress should lead to less M2 state and more M1 state. So we use polyethylene glycol as our osmolyte, which is a hydrophilic polymer, and we use polyethylene glycol with different molecular weight. And the chemical potential of water is directly related to the activity of polyethylene glycol. So when you have a higher concentration of polyethylene glycol, you have a higher osmotic pressure in the system, and we have a lower chemical potential, and then we should expect that it will reduce the, it will dehydrate the protein. And we also assume, when we do the calculation, we also assume the opening of the protein to outside as a Gibbs dividing surface. And we also divide our osmolyte into two groups, large osmolyte and small osmolyte. PEG molecules with molecular weight above 1500 is considered as large osmolyte, and the PEG with molecular weight less than 200 is considered as small osmolyte. So we observe the expected results with large osmolyte where it favored the dehydrated state or meta 1 state, but with small osmolyte we observe the opposite. So I will explain it in the future slides. So to determine the M1 to M2 equilibrium constant, we use a UV spectroscopy and first obtain the your absorbance spectra of the dark state rhodopsin. Then we obtain the absorbance spectra of the light exposed rhodopsin, and then we obtain the difference spectra, and then we fit the difference spectra with the M1 and M2 basis spectra. By using this information, we calculate the fraction of meta 2. And then we plot our data by using the ln K versus osmotic pressure of the system, and was able to derive a thermodynamic relationship between the equilibrium constant between meta 1 and meta 2 and the osmotic pressure of the system, which, and the relationship is quadratic. We also able to calculate the number of water molecules in flux into rhodopsin by using the linear term. And we also able to determine the volume change and the compressibility change of the system during the M1 to M2 transition by using the quadratic term. And as you can see, there, according to our calculation, there is 90 water molecules in flux into meta 1 to results in meta 2. This is largely agree with the molecular dynamics simulation data. And also we have a large increase in our protein volume, and there is a high variability of the protein volume and higher compressibility of the meta 2 state. However, so this observation is only valid with the PEG, which have a higher molecular weight. With a small molecular weight PEGs, our results cannot be fit into a common equation. And what we initially saw, like there is a increasing in equilibrium constant with increase in osmotic pressure, especially you can see it with the PEG 200. So this is directly contradict what we hypothesize that, and we assume that this is maybe due to the fact that the small osmolyte actually penetrate the protein core and somehow stabilize the M2 state. However, after some point, system get saturated and the osmotic effect is start dominating, and we see the expected trend even though they are not fitting into one curve. Then we try to determine this effect of osmotic stress on the pH-dependent rhodopsin conformational dynamics. As you can see from the graph, the lower pH favor the meta 2 state, higher pH favor the meta 1 state, but due to the thermal population, so we don't get like 0% meta 2 even at the very high meta 1 favored condition. Here, there are two key residues in rhodopsin that can deprotonate and protonate during their activation. One is the shift base that is attached to the retinyl ligand. Another one is glutamate 135. So glutamate 135 pKa is directly related to meta 1, meta 2 equilibrium constant. So we can use glutamate pKa value trend to see how the the osmotic stress affect the system. So we conduct experiment and able to derive a relationship. And these results are agree with what we observed with the previous experiments, where higher molecular weight osmolytes favor the dehydrated state, so they will reduce the pKa value of the system. And the smaller osmolyte initially increase the pKa value, so they favor the meta 2 state. Then we calculate the number of water flooded into the system with different PEG molecules, and we were able to find out that their values are consistent with the molecular dynamic simulation, and they are relatively constant around 80 to 100 water molecules. So meta 1 to meta 2 transition always accompanied by flooding of water molecule into the core of the protein is a reliable conclusion. Then we try to figure out whether the hydration, dehydration have a role in the G protein binding, which is required to activation of G protein and the subsequent activation of the effector proteins. To do that, we use transducing alpha C terminal analog rather than using the wild type G protein. And then we generate the binding isotope with different PEG concentrations and different type of PEGs molecules. What we observe is that the small osmolyte doesn't have any effect on the binding affinity of the G protein to the M2 state. However, large osmolyte decrease the binding affinity of the peptide. So this indicates that the large osmolyte dehydrate the protein and reduce the binding affinity of G protein to activated rhodopsin. So both activation process of the rhodopsin and the binding process of the G protein depend on the hydration-dehydration condition in the cell. So based on this information, we develop a new model. We call this.
Hi, Nipuna, if you could please wrap it up. Thank you.
Okay. Call the sponge model, where water dehydrate initial hydration of the system is make it favorable to binding with G protein, and then binding with G protein and M2 state initiate a GTP exchange, and this will result in this will result in dissociation of alpha and gamma, and then cycling of the cycle. So we can say that this new paradigm says that the GPCR activation is go through several steps of dehydration and hydration of protein. So this is a important finding. So the final conclusion we had from our experiments are that during the activation process of rhodopsin, there is 80 to 100 water molecules flooded into the receptor. And this also, we also figured out that decrease in the hydration level will result in decrease in the affinity of rhodopsin to G protein. So we want to expand this experiments to other GPCR, and also we want to test this model with the full-length transducing. These are the people in my group, and they help me with all these experiments. And thank you for listening. And I would like to take a question if you have.
Thank you, Nipuna. Maybe we can fit in one question because we are just running out of time. So one of the audience asked, is the M1 to M2 equilibrium light intensity dependent? So if that reaction is light intensity dependent?
No, actually, so one molecule of rhodopsin absorbs one photon. So then it will undergo conformational changes and then establish the M1 to M2 equilibrium, and it will not depend on the intensity of light. Intensity will determine the number of rhodopsin molecule activated, but it will not determine the equilibrium constant between the M1 and M2.
All right. Yeah, thank you for this nice talk. Some of the audience also compliments on you on that. Yeah, so please feel free to go in and try to answer some other questions there as well. So here, let me introduce our last speaker. So our last speaker is Dr. Blake Hill from Medical College of Wisconsin. And today his talk is Structural Studies of Human FIS1 Reveals a Dynamic Region Importance for DLP1 Recruitment and Mitochondrial Fission. So here, let me hand the time to Blake. So Blake, please go ahead.
All right, thank you, Clement, and thank you all for tuning in, especially those in time zones that make this rather inconvenient. Today, I'm going to share with you some, primarily the graduate work of one student in lab, John Egner, who is looking at the role of the FIS1 arm in recruitment of DRP1 in human cells. As many of you may or may not know, mitochondria are quite dynamic organelles, and they undergo frequent fission and fusion events that are important for function. This has been recognized for well over 100 years now, but the exact role that it plays has been only recently been discovered. And what's come through the work of a lot of people is appreciation of a model in which mitochondrial fission undergoes this fragmentation, the mitochondria undergo fragmentation, and then you have a sort of a quality control checkpoint in which either healthy mitochondria, represented by these darker shades, could undergo and regain, undergo fusion and regain to the healthy pool. And then fragmented mitochondria may be distributed to where they need to be gone, or taken out to the trash through autophagic processes, or if things are really bad, apoptosis. So this is sort of a general thought of the process. And one of the things that my lab's been very interested in is all of these types of mitochondrial fragmentation appear to involve a dynamin-related GTPase DRP1 or DLP1 or DNM1L. All of those are used interchangeably. And yet what dictates whether you have fragmentation for distribution or mitophagy or ultimately apoptosis, all very important processes, is not really quite understood. So what governs these different fragmentation or fission processes is something that we're quite interested in. So fundamentally, how do you go about cutting a membrane? Well, very simply, it's maybe you just need a cutter recruited to a site of scission. In this case, it's a dynamin-related protein, as I mentioned, and it's recruited to a site of scission in lower eukaryotes by a recruiter called FIS1, mitochondrial fission protein 1, which is a resident mitochondrial protein anchored to the outer membrane by a C-terminal transmembrane domain. And curiously, FIS1 is uniformly distributed on the surface of the mitochondria, but DRP1, which is cytoplasmic primarily, is recruited to specific sites of scission. So this suggests that something else is going on here to determine a site of scission. And that's an important question that we and others have been trying to answer for some time now. Ugo Acho in lab, who's a graduate student and one of our bioinformatics experts, has done a sequence alignment, as have others. And really, in looking down at FIS1, what we see here is high conservation through most of the protein. It adopts a common protein-protein interaction motif consistent with the idea that it's a TPR fold. But where it's really divergent is this N-terminal region, which has been dubbed by others as the FIS1 arm. And you see, it's hard to see here, but just basically you can just see very low conservation in the N-terminal region. And so this has led us to, and others, to look at structures of FIS1 and to get a sense of what the protein looks like. And what you see from different species, human on the left, mouse, and yeast, is that the orientation of that divergent region actually varies quite a bit. In the yeast molecule, it actually tucks back into this concave surface that's lined with evolutionarily conserved residues. But in the human molecule, you see the arm does not tuck in to this concave surface. This is a curious feature structurally, but it really was one of the starts of us to look at the N-terminal arm. And we and others have shown that the N-terminal arm plays a very important role in regulation. And the yeast molecule, this is data generated by Megan Harwig in lab, who's shown here with their two adorable daughters. And I'm not gonna, in the interest of time, I won't walk through all of this slide, but suffice it to say that with wild type FIS1, you get normal mitochondrial morphology shown here in the middle, and sort of native recruitment of the yeast DRP1 molecule. And when you remove the N-terminal arm, the first 15 residues, you lose a lot of that recruitment to the mitochondria. You still see DRP1 punctate, and you lose mitochondrial morphology. A quantification is shown here in which expression of DRP1 co-localizes well with mitochondria, and then upon removal of the arm, you lose that localization. So in yeast, it's well understood that the Fis1 arm is necessary for DRP1 recruitment and fission. And the N-terminal region has also been shown a longer sequence by Weerasinghe Yoon's group to also be involved in DRP1 recruitment in humans. So it establishes a really important role for this N-terminal region that has been looked at for quite some time. So why are we continuing to look at that? Well, as it turns out, there's a lot of dispute in the field now on whether Fis1 is actually playing a role in mitochondrial fission. And here I summarize just a short list of a number of different studies. And the data that's against it is that in some Fis1 knockout cells, depending on cell type, you don't have a phenotype suggesting that Fis1 is dispensable. And also in many other eukaryotes and mammals, another resident mitochondrial protein, mitochondrial fission factor of MMF, has been shown to be a dominant recruiter of DRP1 in many cell types. And then in vitro, Fis1 doesn't appear to impact the GTPase activity of DRP1, which is curious if you'd expect it to be regulating its process. So some folks in the field think that Fis1 is dispensable for fission, yet it's one of the two evolutionarily conserved proteins in all species that have mitochondria, Fis1 and DRP1. So the question that we've been asking or testing is whether Fis1 does play some sort of evolutionary role in fission in human cells. For the evolutionary role, what we've been taking is a structural biology approach, and Laura Picton, when she was in lab a while ago, showed in the yeast molecule that the Fis1 arm, or this N-terminal region that I show here, adopts sort of two different conformations. The structure shows a predominantly in conformation, and her work that I'm summarizing in a single slide suggested that it could also adopt this out conformation, and that might be an important regulatory role for the molecule, as I mentioned, because removal of the arm prevents fragmentation and DRP1 recruitment. So the question we had was, is there evidence for this in human Fis1? On the right is the NMR structure solved by Richard Yule's group and Matoshi Suzuki. And on the left is a model in which the arm may tuck in, similar to the mouse structure, which shares 96% sequence identity. That sequence identity suggests that this conformational change could occur in the human molecule. So we asked that question. In fact, John Egner in lab asked that question, and what he first did was to uniformly label a sample of Fis1 with N15 and collect a proton-nitrogen correlation experiment. This is an NMR experiment in which essentially you get one cross peak for every residue in your protein that shows a directly one bonded nitrogen to hydrogen. And what you see in black is a construct of physiological pH that's lacking the transmembrane domain, and then in the blue X's are the chemical shifts for a similar construct that was solved and has the arm extended, as I just showed you. And what you see is there's not overlap here. That means just between these constructs and the conditions in which this was collected, there's been a conformational change. John mapped this using a density kernel plot, and he found that the predominant changes localized to the arm, which would be consistent with an arm and an in-out conformational change. So John then went on to do molecular dynamics simulations, and here's 1,000 nanosecond trajectories, starting with the arm out structure here on the left, and in the middle with the arm in structure. And after 1,000 nanoseconds, you see that the arm is tucked in, similar to what we see in the yeast molecule. This is quantified here, in which these dots are the average structural distances in the NMR structure, and then the bars are the distances in the MD simulations. So the MD basically suggests that there's a possibility, or the potential for the Fis1 arm to be tucked in, similar to the yeast conformation. Is there any experimental evidence that might support that idea? So what what we did then is we looked at the tryptophan, and this is work of MD PhD student in lab, Kelsey Meacham. And what Kelsey basically asked is said, look, this model predicts, given where the tryptophan lies, that if I remove the arm, I should see a difference in the tryptophan fluorescence if the arm is tucked in. And if it's out in solution, there should be no difference between removal of the arm. So she looked at that by tryptophan fluorescence. In blue, you see the native arm, and in red, you see the delta N. And you see a blue shift and increase in intensity consistent with hydrophobic burial in the presence of the arm. That's strong evidence that the arm is actually in a yeast-like conformation. And she also did a Stern-Volmer quenching analysis, which is consistent with this data. So John then went on to ask the same question by NMR spectroscopy, doing this proton-nitrogen correlation experiment with the native arm and the delta N arm. And basically, suffice it to say that what he saw was the largest chemical shift changes localized to the arm and then the region surrounding where tryptophan 40 resides. So these data strongly support the idea that the arm has a possibility to be tucked in. So what does the arm do functionally in human cells? Is it similar to yeast? And here I show work of Megan again, in which in wild type cells, when you have FIS1 knocked out, you see these elongated mitochondria. These are CRISPR-Cas9 knockout cells from retinal pigment epithelial cells that Megan generated. When you overexpress wild type FIS1, you see this fragmentation. And then if you now look at where DRP1 is, you see that you increase the recruitment of DRP1 in a FIS1 dependent manner in these cells. Upon delta N, what you see is you lose your mitochondrial fragmentation and you lose DRP1 recruitment. So the arm is important for fission and for DRP1 localization upon overexpression. And this is all quantified here through a mitograph connectivity score, which allows a quantitative measure of the mitochondrial network. It decreases upon fragmentation and increases upon elongation. And I won't go through all this data except focus your attention on the Pearson's correlation coefficient in the FIS1 knockout cells. This is just all a quantification of what I showed on the previous slide. When you overexpress FIS1, you see an increase in DRP1 co-localization with mitochondria, and you lose that upon removal of the first 8 residues of FIS1. So I've gone through this pretty quickly, but what we wanted to, in the interest of time, I just wanted to share with you these results. And we were interesting to test the hypothesis that human FIS1 might act in a similar manner to yeast FIS1 and have evidence for a regulatory role for the short end terminal arm. And so what I've shared with you is that this region is dynamic. It can adopt an in conformation and an out conformation, and this conformation looks like the yeast conformation, although the arm is much shorter, and it's important in DRP1 recruitment and mitochondrial fission. And one of the things that the lab's very interested in is what governs this conformational change. So with that, I'll thank the people involved. As I mentioned, Megan is our research scientist in lab and the lab manager who keeps us all on the path of straight and narrow, and as an outstanding mitochondrial biologist and expert in confocal microscopy mitochondria. Kelsey Meacham did the tryptophan fluorescence analysis that I shared with you, and Ugo Acho did the sequence alignment is continuing to look at what governs the arm dynamics, and John Egner did most of the work that I shared with you today. So with that, I'd be happy to answer any questions. Thank you.
Thank you, Blake. This is an impressive talk. Indeed, you have like a number of questions. I guess I can ask maybe a few of those. So the first question is FIS1 N-terminal dynamics sensitive to mitochondria membrane damage?
Yeah, that's a really interesting question. We've long thought that maybe the arm is a sensor for something that's happening right at the membrane, be it recruitment of cardiolipin or things like that. And Rob Wells, a student in lab a long time ago, looked at this, and we definitely saw that it was sensitive to membrane curvature. And the experiments that we did with adding then cardiolipin to see if that affected this, it were sort of there was evidence to support it, but it was somewhat inconclusive. That was inconclusive in part because our ability to reconstitute the full-length FIS1 in membrane vesicles has actually been quite challenging and something we're still working on.
Right. Yeah, I think this is a complex system, so I agree. And I think the next questions may be also related to this. So what is the effect of different membrane lipids on FIS1 and DRP1?
Yeah, that's a very good question as well. It's known that DRP1 has a requirement, at least in vitro, for cardiolipin containing membrane vesicles. And so it's thought that a region of DRP1 called the variable domain is a cardiolipin binding domain that aids in getting DRP1 to the mitochondria. Now, what the role of the lipids does, in particular with FIS1, is actually unclear. In work that we've done with Gerard Wong at UCLA, we've shown that DRP1 from yeast can induce negative membrane Gaussian curvature, which is one way that it may be acting in addition to its hydrolytic action as a mechanoenzyme. It looks like it has an intrinsic ability to deform membranes. And of course, in that case, you'd expect that lipids that favor negative curvature would help in that process. And so we don't know the answer to that question, but I think it's very likely that it's involved in some manner.
Mm-hmm. So maybe, well, due to the limit of time, I would just ask one last question. In fact, actually, I still see a number of other questions. See if like you can answer some of the other ones by typing it online. But yeah, let me just ask you one last question. So I think it's also like a very good question. Is there direct interaction between the DRP1 and also the FIS1?
Yeah, thank you for asking that question. It's obviously something that's screaming from my talk here, and I didn't share the data that we have on that issue. Yi-Sung Jung's group showed very early on, as a pioneering study in human FIS1, that there was a direct interaction inside cells through co-immunoprecipitation. I believe that had to be cross-linked, and I don't know to the extent whether they interact in cells without cross-linking has been, I think others have done that. We are just starting to do those co-IPs in our lab. When you add FIS1 to, when you add DRP1 to FIS1 in interaction studies, you know, we've seen different results depending on what the student and lab has done. Generally, we don't see a strong reproducible interaction. But we need to look at that upon removal of the arm and see if that actually impacts that.
All right, very good. Yeah, so like, yeah, that indeed, like questions still coming in. Seems like many people are very interested in your talk. Just, I guess we don't have enough time to just go over them right now. So please feel free to just go and look at these other very good questions. So at the end, I would like to thank everyone for joining this webinar. And I hope you would enjoy our talk.